morning, everybody. Thank you to Dr. Jason LeJeune and Dr. Abby Fish for agreeing to present for this training session. This is an part of our annual summer research program. And usually this is mandatory for all LBRN participants and other REUs to attend lab safety and biosafety training here at LSU. We usually host it at the Life Sciences Annex Auditorium. However, things are different this year, but we are pleased that these two have acceded to our request to continue providing this training session. We right now are at 21 participants. John will, I think, let them in as and when he sees them. In the meanwhile, I'll request Dr. Jason LeJeune to give his training module session on laboratory safety. Dr. Jason LeJeune is from the Office of Environmental Health and Safety here at LSU. Jason, it's all yours.
Thank you, Ramesh. Welcome, everyone. We're going to talk about general laboratory safety here at LSU. But this can be used anywhere you go, any lab you want to go to, because the rules and the thing apply to mostly anywhere. It's for your best protection and safety. So first thing I'm going to do is watch a small video from the Chemical Safety Board, talking about why it's important on university labs to have safety.
Accidents in research laboratories are not isolated events. Since 2001, the CSB has gathered preliminary information on 120 explosions, fires, and chemical releases at university laboratories and other research facilities around the country. The accidents have caused deaths, serious injuries, and extensive property damage. This CSB video reports on 3 laboratory accidents. The 2008 fatality at UCLA, a death by accidental poisoning of a highly regarded Dartmouth College professor in 1997, and a 2010 explosion at Texas Tech University, or TTU, that severely injured a graduate student.
It would be a shame if not every university stopped what it was doing whenever a major accident happens, like at TTU, like at UCLA, and take it as an opportunity to examine their own safety practices to determine what similarities there are.
The CSB investigated the Texas Tech accident, producing a case study that the board believes includes important safety lessons for academia as a whole.
The CSB is concerned with laboratory safety because it's an area that appears, in comparison to industry, pretty unregulated. There is an OSHA laboratory standard, but its focus is on exposure hazards and health hazards of the research work being conducted.
University research is a large and highly competitive enterprise. The CSB estimates there are over 110,000 graduate students and postdoctoral researchers working in academic laboratories across the U.S. Government agencies and private organizations spend billions of dollars sponsoring research at university labs. Academic institutions, principal investigators, and lab workers face significant pressures to innovate, achieve, and gain funding and recognition for their work.
Research conducted at university laboratories is often on the forefront of technology and innovation. It is important that this research continues and thrives, but it must be done within a strong safety culture where preventing hazards is an important value.
Okay, so if you have any questions about those actions they talked about, there's more videos online from the Chemical Safety Board. There's good reports on about what happened. At each one, there were people who had many years of training and experience, okay? And typically that's when something bad is gonna happen. It's either gonna happen when the first time you do it, or the 100th time you do it, because we get complacent and we overlook safety training and things like that. So at LSU, we have pretty good access for training systems online. So if you, and anyone can look at it. You don't have to be from LSU to go on this website and look at it. Okay, we can get you access to look at it. You go to our website that's shown on the screen, go on safety training, access to lab safety training. There's many different things there. Tell you about general safety and specific safety about cryogens, etc. But today we're going to talk more general terms because everyone's at many different locations. So we're going to go in a general term. First, we're going to talk about emergency procedures. That's a lot of things that gets overlooked when it comes to laboratory protocols, is what to do in an emergency. Okay, chemical spills. Okay, we're talking about, spills that go from, you spilling 1 little ounce or 1 little 10 milliliters on a bench top doesn't need to be notified anybody. Clean it up and move on. We're talking about, liters or 500 milliliters or bigger type spills, or something really nasty you spill, or something really deadly or toxic or bad you spill. Okay, always notify someone superior to you, your grad student, okay, the postdoc, supervisor, the PI. Okay, if your lab group and your PI cannot handle the spill or is not comfortable handling the spill, call your local EHS department or campus police wherever you're at. And campus police should be 911 no matter what university or location you're at from a campus home. But importantly, protect yourself and others from exposure. So if you drop something and it starts fuming and it smells terrible, get out. Okay? Get everyone out, close the door, call people, call your PI, call EHS. If it gets really bad, pull the fire alarm, evacuate people from the building. Okay? If it's flammable, if you can, turn off any ignition sources that you have in the lab. Bunsen burners, hot plates, etc. Okay? Again, begin evacuation necessary. Always follow directions. Never go in or out of the building unless absolutely you're given permission to do so. Okay? Again, spill response. Lab personnel. Okay? We want you in your lab to clean up 99% of what you spill. Okay? If you think it's unsafe or it's too big for you, you call your EHS next. LSU, that's our number. But whatever university or building you're at, look up their health and safety department and find their number to help you. Okay? Facility services, if it's some kind of other but chemical spill. If a pipe bursts in your lab, find out who the facility services are in your lab. Or if a light burns out or something like that, find who they are. Learn their number, learn how to contact them. The last case is going to be city fire and hazmat. That's a worst case scenario. Okay? That's if the fire alarm goes off and the sprinkler comes on, or we evacuate the whole building, then we got to get hazmat out. Okay? That very rarely happens, if it all happens. Okay? To help you recognize spills, level 1 can be handled by lab workers. Okay? Level 2 can be handled by EHS. Level 3 can be handled both by EHS and local hazmat. Okay? Level 3 comes in that EHS will decide whether we need hazmat or not, because at least at LSU, I can't claim for other universities and other buildings, but we are hazmat trained in our department, so we can handle hazmat spills. Okay? Level 4 is hopefully you never reach level 1. Okay. Okay. We have an issue with the recording.
We're back. We're back.
Good.
Sorry about that. I don't know why it stopped. One of us.
So level 4, okay, if you have level 4, the entire building will probably be evacuated. Fire department will show up. State or local hazmat will show up. State in Louisiana, the state police probably will show up, and that's a very bad. Also will show up with the news crews and news containers, and then it's going to be bad. But if we have to do it, we will do it, okay? And no, for any of you going into the lab, think of this as new eyes. So look around. If you see containers that are bad, please note it. Please tell somebody, okay? Because at least once a year, we have to get this bomb squad to come out and remove containers from labs because they're so old and they're hidden behind a cabinet somewhere. And even as a chemist, I'm afraid to touch them. So then, please, use some caution discretion. Okay, injuries. Determine the extent of injury. Okay, if it's a cut If it's bleeding, learn how to treat it. First aid, okay? Make sure you know some kind of first aid treatment, some CPR. You never know what could happen. Someone could pass out. Someone could collapse in the lab. It happens more often than you would think, okay? This is obvious, but avoid contact with all body fluids, especially now in the COVID area. Be very vigilant. Call 911 if needed, okay? Don't run away and panic. Stay to assist emergency personnel. That doesn't mean you have to provide medical care, but you were there and you saw what happened, so they will need to know what happened. So please stay there and tell emergency personnel what happened and let them release you, okay? And always, if there's an accident, secure the scene. Don't move things around unless it's a safety issue, so we can do investigation and figure out what happened and what went wrong. Okay, fires and alarm. Okay, always remain calm. You're not a firefighter, so don't go try to put out a fire, okay, unless you've been properly trained. Okay, if the fire's anywhere but inside of a hood, pull the fire alarm and begin evacuating the building, basically. Okay, at LSU, you call campus police. 911 from a landline. If not, you call 225-578-3231 from a cell phone. Okay, always follow directions. If you're in a building and the fire alarm goes off, you must exit the building. You can't say it doesn't relate to me. It doesn't mean I have to leave. No, you have to leave. Okay, too many times the fire alarm goes off in a building and we have to go in floor by floor and search to make sure everyone's out. Okay, so that means keep your ears open so you can hear what's going on around you. Don't put these Beats headphones on and like this, you can't hear anything going around you, okay? Because the lab across could blow up and you're going to be staying there and the last one out and you could be stuck. Okay, and don't go back in until someone, police or someone in authority gives you permission to do so. Okay, with radiation emergencies, okay, if you're going to deal with radiation, get familiar with the radiation safety in your location. Okay, get special training from them if you want, but notify radiation safety at LSU or wherever you're at. Keep all contaminated personnel in general vicinity. We don't want someone who has some kind of radioactive material on them to go touch everything down the hallway. Okay, then we have to decon everything down the hallway they touch. So keep them, you know, you can move them from the lab into an office or something, but keep them there. Okay, secure the lab. Don't let anyone back in after a spill occurs. The radiation safety comes in and gives you the clear. Okay. With fires for radiation safety, evacuate. Don't try to put out. They're too dangerous. Okay. You don't know what's burning exactly and what kind of molecules we put in the air. Okay. Very bad. For injury, handle the same as a non-radiation injury. Okay. Treat it, evacuate them, notify radiation safety officer. Okay, PPE, very important, especially in today's world. Okay, eye protection, safety glasses must be worn at all times in the lab. Okay, if you're messing with any kind of liquid, especially, safety glasses must be worn. Okay, if you're messing with any kind of hazard that will fly into your eye, like if you're cutting glass or using needles, safety glasses must be worn. Okay, safety goggles must be worn when using high hazardous materials. Something like acids and things like that, okay? Because glasses won't protect you 100% of the time from splashes. We had several incidences in chemistry at LSU where people were wearing safety glasses and still got chemicals in their eyes, okay? So it's possible if you wear safety glasses, chemicals in your eyes. Okay, face shield. Again, face shields are to be used, again, with some high hazard like acids, okay? You can use goggles for acids, but if you want to protect your whole face, use the goggles plus a face shield so it doesn't splash anywhere in your face. Now, with a face shield on, you're wearing a lab coat, you're fully protected from any kind of splash that will happen to you. Okay, now gloves. When it comes to gloves, please pick the gloves that are safe for you to use. Know if you're allergic to latex or not. Okay, notice if the type of glove you use is compatible with the chemical you use. A good example I like to use is chloroform. Chloroform and nitrile gloves are not compatible. Chloroform will dissolve nitrile gloves, okay? And dichloromethane and chloroform will dissolve nitrile gloves. So please, look at the chemical compatibility charts on gloves you use and what type of gloves you need to use. Protective clothing, okay? Lab coats or aprons should be worn at all times and in the lab, okay? Must be splash hazard if you're going to mess with corrosives, etc. Okay? Flame resistant lab coats should be used if you're going to mess with fire hazards. Okay? If you've ever been on LSU campus, if you have typically have a blue lab coat, that's flame resistant. A white lab coat is not flame resistant. Okay? So wherever you go, get the lab coat that's going to be pertaining to your level of research. If it's bio, get a bio lab coat. If it's chemical, get a chemical resistant lab coat. If it's going to be flame resistant, get a flame resistant lab coat. Or get a flame resistant and chemical resistant lab coat. Okay? But learn something about where you're going, what you're working with to protect you from the hazards. Footwear. Okay? Open-toed shoes and sandals are never allowed to be worn in the lab. Okay? Always do closed-toe. And typically, if you're going to be using hazardous liquids, don't use running shoes or tennis shoes, something like that, okay? Why we don't want to use those? Because they're good for running. They breathe, okay? Any running shoe that gets wet, your socks are going to get wet. So the last thing you're going to do is spill sulfuric acid on your running shoes and gets on your socks. Well, then you have capture there. So you have a gap that's going to capture the sulfuric acid in between the socks and the shoe, and it's going to burn you very badly. Versus, fully leather-toed shoes, etc. Okay, this is only when you're using high hazardous materials or liquids you don't want to get on your skin. You use that. For any other times, just closed-toe shoes, any kind of closed-toe shoes would be allowed. Okay, so this is emergency numbers only for LSU campus. Again, I encourage you, please look up the emergency numbers on whatever campus you'll be working with. Even if you're working virtually, it's good to know the safety numbers of the campuses you'll be with, okay? Imagine yourself in the lab and what you'll be doing, okay? And so always know the area code, too, for the area you're working in. In Baton Rouge, it's 225. Other places, it could be different. Okay, so now we get to the regulatory issue parts of safety. Everything you create must be labeled, okay? The name of the material label must match MSDS if you have an MSDS or SDS, okay? So if you pour out ethanol from the ethanol container into a bottle, the label on the bottle has to match what the label is on the ethanol. So you can't call it something that's not commonly known, okay? It must have specific hazards on it if you know them, right? Physical and health hazards. So if something's highly flammable, you need to know that. If something's carcinogenic, you need to know that. If something's heterogenic, we need to know that. If something's mutagenic, we need to know that. Okay? So the last thing you want is to have 50 samples on your desk with no names on them, and something happens, and we go in, and we're the ones that got to tell the police and the fire if it's safe or not. Well, if there's no name on it, I don't know. Abby won't know. Nobody's going to know. So we're just guessing. So we got to take full precaution. So it's both for our safety and your safety that we know everything must be labeled. Okay? Chemical safety. I can't say this enough to people. Know what you're doing when you do it. It seems pretty simple, but you'd be surprised how many grad students we come across that don't fully understand what they're doing and what they're working with. I've had many cases that they call me in the office, this chemical is doing something in my hood, what should I do? I'm in another building, I don't see what you're working with, so how in the world would I know what to do? Okay, but that's the kind of questions we get, and that's the kind of questions that really annoy me. You are the expertise in what you're doing, not me, okay? I'm there to offer you consultation and help you, but I'm not there to solve your problems for you, okay? So know what to do in day-to-day operations and emergencies. Where proper PPE required, or when exposed potential exists. Know the emergency procedures. Like if there's a firearm, where do you go? How to get rid of stuff. Okay, how to get rid of gloves, how to get clean lab coats, how to clean glasses, etc. Okay, know the locations of safety showers. Hey, make sure they're tested and they work. Okay, of eye washes, test them if they're plumbed in. The last thing you want is to turn on eye wash and it comes out brown. That's rust in the water. You don't want to get rust in your eye when you're trying to use it when it's important. So again, test those, clean those, make sure they're safe to use. If you notice signs of overexposure, that could be physical overexposure, chemical overexposure. Leave the area, get fresh air, and swim and help. And most importantly, especially now, wash your hands frequently and before eating or taking a break. Okay? This were rules before COVID, but it's really important now after COVID. Okay, so a little thing we put in about when laboratory operations come back in the COVID world, what you could look forward to. Okay, this is general recommendations, not rules yet. We're working on that. Always maintain 6 feet separation if you can. Okay, if not possible, wear lab coats, gloves, and face masks. Clean, sanitize hands often and always when leaving lab. Work in staggered times or days if possible. Okay, do not report if showing symptoms of COVID. Do not report test positive for COVID, and always follow guidance at the university. Okay, each university will have guidance and guidelines how what to follow. So make sure you follow them. Okay, and that's it. Always safety first, and I'll be happy to answer any questions.
Awesome. Thank you. Anybody has any questions, you're welcome to ask Jason right now, or if something crops up in your mind, perhaps you can email it to John. Or
Actually, Ramesh will have a link afterwards with the recording, and Jason has offered his PowerPoint, so they'll be able to ask it in the form there instead of emailing me directly.
awesome. And we also have last week RCR talk available for another 3 or 4 days, and we'll have Jason's training, and a little while later Dr. Fish's training available for later viewing for those who are unable to attend today or who still want to refer back to it later. So let us know if you have any questions. We'll give you a minute or two and you can unmute and ask. Otherwise, we'll move on to Dr. Abby Fish. No questions popping up, John?
No, you guys can put it in the chat as well. All right. Well,
All right, let's move on to the next talk. It'll be by Dr. Abby Fish. She's the Biosafety Officer here at LSU. She's again also with the Environmental Health and Safety at LSU, and it's a natural transition initially to learn about primarily lab safety, and then we are the Louisiana Biomedical Research Network. It's a natural transition to learn about biosafety, and I'm thankful for Dr. Fish to provide us this training. Abby, it's all yours.
Okay. Good morning, everybody. Thank you for that introduction, Ramesh. My name is Abby Fish, and I'm the Biosafety Officer here at LSU, and I work with the Office of Environmental Health and Safety. And today I'm going to be discussing with you just basic laboratory. So before we get started, I want to define 2 terms for you. The first being biosafety, and what that is defined as is reducing or eliminating exposure of individuals and the environment to potentially hazardous biological agents. Biosecurity is another important term, and it's also related to biosafety, and it is defined as protection of microbial agents from loss, theft, diversion, or intentional misuse. So biosafety is achieved by implementing various degrees of control and containment. And that starts with the design of the laboratory. It's also important to restrict access of the lab to individuals who work in this space. Personnel experience and training is also a major part of biosafety. We want to make sure that everyone in the lab knows what they're doing. And then there's also specialized equipment and safe methods of managing infectious agents. Those are also two big parts of biosafety. So biosecurity is achieved by limiting access to these research facilities, the materials, and then the information that's related to both the lab and the facility. So biosafety and biosecurity share a lot of common components. Both are based on a risk assessment, and that's going to be a risk assessment just kind of assesses the different hazards that are associated with the lab, the types of procedures, and then the hazard itself, you know, how infectious is that to humans? Is it a plant pathogen? Is it an animal pathogen? What kind of environmental risk does that have? It's also going to be based on personnel expertise and responsibility, control and accountability of the research materials, and then access control to the lab, material transfer documentation. And basically what that means is, do you have all of the proper permissions to be working with the things that you're working with? Training is important, and then also emergency planning and program management. So the CDC and the NIH have established a set of guidelines that help individuals in a lab recognize the hazards of the biological agents that they're working with. So these regulations outline the precautions and special practices and decontamination procedures that are required for a specific agent. They are in place to protect workers in both microbiological and medical labs through the use of engineering controls, which we'll talk about here in just a minute, standard operating procedures, and then work practice controls. So complete information and recommendations can be found within the Biosafety and Microbiological and Biomedicals Laboratories petition, which is what's pictured on the right-hand side of your screen. And then also the NIH has established guidelines for research involving recombinant or synthetic nucleic acid molecules. There's two links provided here that you can get the full, battery of information on all of that. So one of the major ways that we protect ourselves in a lab is going to be through the use of primary barriers and universal precautions. Primary barriers are physical barriers or personal protective equipment that are physically put between a person and the hazardous agent. Examples of this would be biosafety cabinets, specialized breathing apparatuses such as an N95 mask or a cloth mask, or a personal air purifying respirator, safety glasses, lab coats, or safety caps on centrifuge buckets. Universal precautions are a set of guidelines that are aimed at preventing transmission of bloodborne pathogens from the blood and other potentially infectious material of one to another. And these guidelines were originally developed in the 80s in response to the outbreak of HIV and were originally aimed at protecting healthcare workers. Now, they're still very applicable to microbiology labs because they provide us guidance on how to handle labs or during an outbreak investigation, such as right now, what we're experiencing with COVID-19. So universal precautions are going to include things like washing your hands thoroughly, using disposable gloves, wearing lab coats, potentially wearing disposable gowns or face masks, the use of face shields, safety glasses or goggles. And then also universal precautions are going to dictate how we properly handle sharps, such as needles and scalpels. These guidelines require that all potentially infectious material and contaminated equipment be handled in a manner in which to prevent potential contamination or transfer of infectious material. So there are special circumstances in which additional precautions may need to be taken. So that would include additional protective equipment. And right now, one of the big things we're experiencing back to COVID-19 is if you go to the hospital, they're giving you a mask to put on. And if you end up admitted into the hospital, the doctors and nurses are going to come into the room wearing additional PPE than what they would traditionally have been in the room wearing. So when working in a lab, all personnel should have adherence to standard microbiological practices and techniques. All individuals need to be aware of the potential hazards that are associated with working in that specific lab. You also need to be properly trained and proficient in the safe practices and techniques of that particular lab. And all of these are going to be different depending on the lab you're working with and the hazards that are associated with that specific lab. The responsible scientists for each lab will be responsible for providing the appropriate training. And this could be, a lab manager, the lead scientist, it could be the PI that you're working with, or it could be, a graduate student or a postdoc that is proficient in these techniques. So in addition to the specialized training that you will receive in each lab should also have a laboratory biosafety manual that is developed specific to the hazards of that lab. It will need to include any special practices and procedures to minimize or eliminate your risk of exposure to that hazard in that lab. All personnel that are in the lab are required to read the biosafety manual and are responsible for all of the work that is being, or for the work that is being conducted in that lab. This sounds harsh when I say this, but I didn't know is never a good excuse. You need to be trained and knowledgeable in the specific techniques, safety procedures, et cetera, that are associated with the work you're doing. So the biosafety and microbiological and biomedical labs, the BMBL is what we call it, is the basis for developing this manual, but it should not be used in its place. So in addition to that manual, each lab will have specific safety equipment that has been designated for use in that lab. The equipment is going to be used to contain splashes, splatters, aerosols, any type of potentially hazardous droplet that is generated during manipulation of these agents. So safety equipment is one form of what we call an engineering control. And engineering controls are used to protect lab workers from biological hazards by placing a physical barrier between that person and the hazard. The controls not only individuals, but they also protect the community and the environment from the hazards. And then, some engineering controls, such as a biosafety cabinet, protect the material from becoming, contaminated from the outside environment. Safety equipment for all biosafety levels will include biosafety cabinets, enclosed containers, and then safety caps on centrifuge buckets. So personal protective equipment is another form of safety equipment. Proper personal protective equipment, or as I'm going to call it from now on, PPE, will include a combination of disposable gloves, a lab coat, or a disposable gown, sleeve covers, closed-toed shoes, potentially shoe covers. Could also include a respirator or a face shield, safety goggles or safety glasses. And the required PPE will vary depending on the level of work, the amount of hazardous material that is being used, and the type of biological agents that are used in that laboratory. So PPE is generally used in conjunction with biosafety cabinets, but the requirements are different for each biosafety level. So the second major part of biosafety is what we call secondary barriers. And secondary barriers describe the requirements for the design and construction of a laboratory facility. A good design contributes to the overall protection of the lab workers, the individuals outside of the lab, and it also protects the environment and community from the hazards associated. So the recommended secondary barriers depend on a couple of things, including the risk of transmission of the agents in the lab. Higher levels of containment are going to be required for more hazardous biological agents. And some examples of recommended barriers include sinks for hand washing, eye wash stations, safety showers. Some facilities have specialized decontamination facilities such as autoclaves. or specialized ventilation systems. So labs are divided into 4 biosafety levels, each of which consists of a combination of specialized lab practices and techniques, safety equipment, and then lab facilities. The protective practices increase with each level and build upon the practices established by the safety level below it. So BSL-1 levels or BSL-1 labs are going to be are going to work with the least dangerous agents, and they're going to require the fewest precautions, whereas a BSL-4 lab is going to work with more the most dangerous agents and have the strictest precautions. So the assignment of a biosafety level to a lab dictates the proper laboratory practices and techniques, the safety equipment that needs to be used, and then also the design of that facility. Designation is determined by a risk assessment, and it depends upon several factors, which include the biologic agents that are going to be used, as well as the route of transmission of this agent. Ultimately, the appropriate level will be determined by appropriate staff, which will include the bio officer and the institutional biosafety review committee. So BSL-1s are the lowest level lab, and they are designed for educational training and teaching labs. This designation is generally used for work involving the use of well-defined and characterized agents that are not known to cause disease in healthy humans. Some examples of a BSL-1 agent would be Bacillus subtilis, Naegleria gruberi, and then any organisms that are exempt under NIH guidelines. What you see pictured on your slide, the top is going to be a picture of the Bacillus subtilis, and the bottom is a picture of Naegleria gruberi. So BSL-1 labs have the most basic level of containment, and they rely on standard microbiological practices with no special primary or secondary barriers required apart from hand washing sinks. BSL-2s are designed for research, clinical, diagnostic, and teaching, as well as other laboratories that work with a broad spectrum of indigenous agents. These agents are going to be moderate risk agents that are present in the community and are associated with varying levels of human disease. Work can be conducted on an open bench with good standard microbiological techniques, providing that the risk for splashes and aerosols is low. The use of a biosafety cabinet is required for work that involves potentially infectious materials such as human or primate cell lines. And that's just to protect the individual from potential exposure. So some examples of BSL-2 agents would be B virus, HIV, Mycobacterium leprae, Salmonella typhimurium, and then primate or human derived blood, body fluids, tissues, or cell lines where the presence of an infectious agent is unknown. So the primary hazards to individuals that work in a BSL-2 is going to be related to accidental percutaneous or mucous membrane exposures or ingestion of the infectious material. Extreme caution needs to be taken when working with sharps. So even though these organisms that are used in a BSL-2 are routinely manipulated at that level and are not generally known to be transmissible by aerosols, extreme caution does need to be taken with procedures such as vortexing that are going to generate a lot of aerosols. Required personal protective equipment includes gloves, a lab coat, closed-toed shoes, and safety glasses. And then biosafety cabinets are required when working with the potentially infectious materials to reduce the probability of contacting aerosols and splashes. So the majority of labs at LSU are going to be BSL-1 and BSL-2 labs. Now we're talking about some of the higher level containment labs. So the first one we'll talk about is a BSL-3. These are designed for clinical, teaching, research, or production facilities that work with indigenous or exotic agents. with potential for respiratory transmission, and agents that are used at BSL-3 may cause serious or potentially lethal infection. Some examples of that is going to be Mycobacterium tuberculosis, St. Louis encephalitis, which is a virus, and then Coxiella burnetii. Primary hazards to people that work in a BSL-3 is going to be exposure to infectious aerosols, auto-inoculation, and ingestion. Laboratory personnel should use primary and secondary barriers to protect themselves, the community, and the environment. And the primary hazard that you're going to be looking for in a BSL-3 is exposure to infectious aerosols. So at this level, more emphasis is put on the primary and secondary barriers than BSL-1 and BSL-2. And that's just to protect you from contagious areas. Biosafety cabinets are going to be required for all manipulations of the agent, and additional personal protective equipment, including disposable outerwear and shoe covers, double gloves, and either a PAPR, which is personal air purifying respirator, or an N95 is going to be required. The access to the lab is going to be tightly controlled, and it's going to be limited to individuals who've had extensive classroom and hands-on training. Some of the secondary barriers you're going to have in this lab is going to be HEPA filtered directional airflow out of the lab to prevent recirculation of contaminated air. And then additionally, you're going to have specialized decontamination facilities. Just as a side note, there is a BSL-3 on LSU's campus. So now we're going to talk about what we call the high containment lab. BSL-4s are required for lab work involving dangerous or exotic agents that pose a high risk to an individual of contracting a life-threatening disease via the aerosol route of transmission, for which there is no preventable vaccine or therapeutic. BSL-4s are also required when working with agents, related agents of unknown risk of transmission. And examples of BSL-4 agents is going to be Ebola virus or Marburg virus. The primary hazards of working in this lab is going to be the respiratory exposure to infectious aerosols. There's also potential for mucous membrane or broken skin exposure to infectious droplets, and then also auto-inoculation. All manipulations within a BSL-4 pose a high risk to lab personnel, the community, and the environment, and therefore primary and secondary barriers are incredibly important. So complete isolation from aerosolized BSL-4 agents is achieved by wearing, you look at the right-hand side of your screen here, it is a full body air supplied positive pressure suit. And you can kind of see at the top where he has the little hose hooked up to his suit where he's actually getting outside air. So BSL-4 is also an access controlled lab that is generally located in a completely separate building or in an isolated area of a building, and only personnel who have received many, many hours of classroom and hands-on training have access to this particular lab. They also have highly complex secondary barriers in place, including specialized ventilation system and a specialized method of disposing of waste. There are no BSL-4 on LSU's campus. So pictured here is just going to be a summary of what we've just discussed and the minimum requirements of each biosafety level. Additional details can be found within the BMBL if you are so interested. So we won't spend too much time on this slide. So in addition to the biosafety level designations, there are a couple other containment categories that are utilized for work with animals or plants or arthropods or recombinant DNA. Each of these is going to have its own categories designated 1 through 4, with 1 being the least hazardous and 4 being the most. So as the biosafety officer at LSU, part of my job is to inspect all labs on an annual basis for compliance with federal regulations and guidelines. And during these inspections, I'm going to assess for the presence of the biosafety manual and a copy of all standard operating procedures. I'm also going to be looking for documentation of proper training of laboratory staff. I also look for appropriate postings and labelings for hazardous material in the lab, as well as safety materials such as safety showers, eye washes, first aid kits. I'm also going to be examining the staff for the use of proper PPE, as well as access to that PPE. The laboratory design and setup is sometimes out of the investigator's control, but we do like to make sure that the individuals have everything they need to properly contain the hazards that are associated with the lab. And then lastly, I look for proper disposal of waste. So pictured here is a couple examples of the door postings. Door postings are utilized outside of each lab to communicate the hazards that are associated with that lab. And that information needs to include the biosafety level of each lab, the agents that are being studied, and then also the contact information for the responsible parties, including emergency contact information. All right, so just to summarize what we've talked about a little bit today, research should be evaluated from a biosafety as well as a biosecurity standpoint. Guidelines have been developed to protect workers in all types of labs through a combination of safeguards, including engineering controls, management policies, and work practices. The differences between biosafety levels dictates the necessary precautions for each level of work. And when in doubt, refer back to the BMBL, as that book has a pretty thorough, pretty comprehensive knowledge of how to run a lab at all 4 safety levels. And with that, I will take any questions.
All right, open for questions. Anybody has any comments, questions for Dr. Fish? Dr. Fish mentioned we have a BSL-3 facility here at LSU. And she mentioned some of the pathogens that are usually associated with BSL-2 use. One of the pathogens is the COVID-19 that were clinical samples that were handled in our BSL-3 facility. My apologies. Yeah, and there is obviously somebody. Dedra McKee made a comment saying very informative. Yeah. So anybody else has any comments, questions for Dr. Fish? These talks will also be available. They are recorded and will be available on the LBRN website. And I realize that right now we are all in a virtual setting, and active laboratory work is some distance away. But we hope that as and when this COVID scare passes, or there is appropriate treatment available, then we will resume active lab work. And should this thing happen in the next 6 months or so, there is opportunity for some of you summer research participants to participate in the Summer Scholars Program. That will be an immersive lab experience. So this is a good idea for you to be aware of the necessary safety precautions with lab safety and biosafety. And these recordings are available, so you can go back to revisit these talks, these training sessions at a later time to acquaint yourself of all the necessary precautions. And somebody wants, Roxanne Shamcho wants the PowerPoint. I think it will be made available.
Yeah, so Jason has already sent me his. Abby, I believe she'll send me hers as well. I'll provide it with the link. And in contrast to the former, or the previous, the RCR recording, which is only a limited time available, I don't think we have a time issue on this one. The reason why the other one had a limited time is because of the ethical discussions. Dr. Penn didn't want that recording to stay active. But this one will be, like Ramesh said, available as long as we need it until the next time.
Indeed.
With Jason and Abby's permission, we appreciate that.
Thank you, both of you. And I don't see any messages or comments, questions popping up. So in which phase in LA will research labs be operational? Jolyn, I sent you a reply earlier today. There is, this is a very subjective question because it depends on university policies, it depends on institutional policies and NIH policies. So this will be a while in the making. We don't know yet. The phases would apply to students within the institution. When people want to bring in students from other places to do research, it can get tricky. So we will keep you appraised and informed, but we do not envisage a summer research opportunity, summer in-person, in-lab research opportunity this summer. Hopefully, maybe late fall or in spring, if things have settled down. We might extend the summer scholars program to some of you to avail the in-lab experience and continue with the project that you're working on right now or something new. But we do not envisage this to happen in the immediate future. All right, anybody else? Okay, if I don't see any other questions, then we'd like to once again thank Jason and Abby for taking time out to give this training session for all of us. As mentioned before, these recordings are available, will be available on the LBRN website. If you have any questions, there is a link there. You can add your questions to that link, and Dr. Fish and LeJeune will be, I'd like to believe, happy to respond, and I will comment on their behalf. And we will try to answer your questions as best we can. With that, I'd like to say goodbye. You all have a nice week and stay safe, everybody. Bye-bye.